The quest to understand the biological underpinnings of human longevity has increasingly focused on the role of genetics, especially in individuals who live well beyond the average lifespan while remaining relatively free of chronic diseases. Although lifestyle and environment are undeniably crucial, specific genetic variants appear to provide a biological edge, offering resistance against the wear and tear of aging, particularly in the realms of cardiovascular, metabolic, and neurodegenerative health. Among the most studied genes in this context are ApoE2, FOXO3A, and SIRT6, each of which contributes uniquely to the cellular mechanisms that influence aging and resilience. One of the most well-characterized genes in the context of aging is APO, best known for its role in lipid metabolism and its powerful influence on Alzheimer's disease risk. While the ApoE4 variant increases the risk of neurodegeneration and cardiovascular disease, the E2 variant is associated with protective effects. Individuals with the ApoE2 allele tend to have lower levels of LDL cholesterol, reduced inflammation, and delayed onset of age-related cognitive decline. This variant may also influence the clearance of amyloid beta from the brain, thereby reducing one of the major pathological hallmarks of Alzheimer's. The fact that ApoE2 is more common among centenarians than in the general population underscores its potential role in promoting both lifespan and health span. Another key player in longevity is FOXO3A, a transcription factor involved in oxidative stress response, DNA repair, insulin signaling, and apoptosis. This gene acts as a master regulator of cellular homeostasis, and its function is conserved across species, from worms to humans. Longevity-associated variants of FOXO3A appear to enhance its activity, allowing for more efficient cellular maintenance and greater resistance to age-related damage. In human studies, individuals with certain FOXO3A polymorphisms are significantly more likely to live to 100 or beyond, and to do so without developing common age-related diseases. This gene's influence on insulin sensitivity and inflammation is particularly important, as both are central pathways in the aging process. SIRT6 belongs to the family of sirtuins, proteins that are closely linked to DNA repair, telomere maintenance, and metabolic regulation. Among the sirtuins, SIRT6 has emerged as a standout for its role in maintaining genomic stability and suppressing tumor growth. It facilitates the repair of double-strand breaks in DNA and modulates glucose metabolism in a way that supports longevity. Mouse models overexpressing SIRT6 have shown increased lifespan and improved resistance to metabolic disorders and age-related decline. In humans, rare but functionally advantageous variants of SIRT6 may contribute to the exceptional longevity observed in some individuals, particularly by protecting against diabetes and cancer. Although these genetic variants are not determinative on their own, they illustrate how specific biological pathways, lipid transport, stress resistance, metabolic regulation, and DNA repair collectively shape the aging trajectory. Importantly, these genes do not operate in isolation. Their expression and impact are modulated by environmental factors such as diet, physical activity, and exposure to stressors. This interaction between genes and environment explains why individuals with similar genetic profiles may experience very different aging outcomes, depending on their lifestyle. As genetic sequencing becomes more accessible, researchers are beginning to map polygenic longevity scores, which aggregate the small effects of multiple genes associated with long life. These efforts aim not only to identify those predisposed to longevity, but also to uncover new therapeutic targets. For example, pharmaceutical research is exploring sirtuin-activating compounds, FOXO3A, enhancing molecules, and apomodulating therapies as ways to mimic the natural advantages seen in long-lived individuals. Understanding the longevity genome also has implications beyond individual health. It challenges existing models of aging by suggesting that decline is not inevitable, but rather a process that can be decelerated with the right molecular tools. Moreover, these insights invite a shift in preventive medicine, from treating diseases after they emerge to proactively maintaining cellular function and resilience. In the broader picture, genes like ApoE2, FOXO3A, and SIRT6 represent more than just rare biological gifts. They serve as roadmaps for designing interventions that extend health span across the population. They exemplify how evolutionarily conserved mechanisms can be harnessed to delay disease onset and preserve function well into old age. While we may not all carry the longevity genes of centenarians, decoding their secrets brings us closer to strategies that support healthy aging for everyone.